Okay, so thank you very much for the introduction. Hello, my name is Francisco Alonso. I come from the Autonomous University of Madrid, and I'm going to present the work synthesis of magnetic nanoparticles using rapid microwave plasma hydrogenation for biomedical applications. That is a work that we have performed at the Autonomous University of Madrid in Spain in collaboration with the Universidad de Porto in Portugal. So first of all, why it's important to do research in this field in magnetic nanostructures for biomedical applications? Well, magnetic nanostructures have several applications in medicine that can go from diagnosis applications. For example, it's interesting to do research in magnetic nanostructures to be used as new magnetic resonance imaging contrast agent, and also for biomolecule separation. And also, magnetic nanostructures have multiple applications for therapeutic purposes. For example, they can be used to treat cancer by magnetic hyperthermia, to deliver drugs in a specific part of the body by applying a magnetic field in that part of the body, and also, for example, in cellular magnetic stimulation. That is the application that we are going to use as a proof of concept of our nanoparticles. It's been reported in previous works that by modifying the size and the shape of our nanoparticles, we can modify the biodistribution of the nanoparticles among the different organs of the body. So it's interesting to be able to control these parameters, the size of the shape of the nanoparticles. And also, it has been reported that for non-spherical nanoparticles, for example, for nanotubes or discs, since they have a higher specific surface area, they are going to be more likely to construct cells since they have more contact points. And also, we are going to have an increased circulation of the nanoparticles through the body. Also, when designing or when synthesizing magnetic nanoparticles, it is always desirable to have the highest saturation magnetization as possible, since in this way we are going to be able to cause the same effect, but by applying a lower concentration of nanoparticles, and this is going to reduce the toxicity of the treatment. And also, it's desirable to have low causativity and remanence, and of course, the distribution we are going to want it to be as monodispersed as possible. So the first part of the work consisted of the synthesis of hematite nanoparticles by an hydrothermal synthesis procedure. We used a 200 milliliters reactor. The process was at 220 degrees for 24 hours. And we used as precursors iron chloride and ammonium phosphate. And what we found here is that by changing the ratio between both precursors, we can control the size and shape of the nanoparticles. Here we have the scanning electron microscopy images of the nanoparticles. We can see how when we don't add any phosphate, we obtain nanoparticles that seem to be nanocubes, and that are more or less 400 nanometers in size. If we add some phosphate, the nanoparticles we obtain are larger, more than 1 micron in length, and they seem to be like ellipses. And if we double the content of phosphate, we obtain like more sharp nanoparticles. We have called them nanoneedles, and the size is reduced a little bit. We perform some X-ray characterization, and we see that, as expected, the diffractogram corresponds to the hematite, and we don't observe important variations in the crystalline size of the different nanoparticles for the nanocubes, nanoliths, and nanoneedles. So up to this point, we have been able to synthesize hematite nanoparticles with controlled shape and size. However, we have to reduce them to iron or to magnetite in order to make them magnetic. we follow two approaches. The first one is like a more common reduction process, that is a thermal reduction process that we performed at 480 degrees for 5 hours in a mixed atmosphere of hydrogen and argon. This procedure has the disadvantage that it's very long. It takes a lot of time. 5 hours plus making vacuum, etc. It takes almost 9 or 10 hours. So it is not efficient to be scaled up for industrial application. However, here we have the results we obtain with this method. We see how by applying this thermal annealing process, the shape of the nanoparticle doesn't change and the size. However, we see like an increased porosity that is especially important for the nanoneedle sample, as we can see here. And we observe how for the nanocubes and the nanoneedles, almost all the hematite has been completely reduced to iron, with only about 4% of magnetite. We don't have any hematite. However, for the nanolipses, we see how we only have about 60% of iron and the rest is still magnetite. We think that the worst reduction of this sample can be because of its larger size, and it's more round, the nanoparticle, and larger. So maybe the hydrogen has not been able to penetrate through all the bulk of the sample, and that can be a plausible explanation. We perform some magnetic characterization, and we can see how the two samples that were almost completely reduced, the nanocubes and the nanoneedles, have saturation magnetization higher than 200 mg. However, the nanoellipsis sample have saturation magnetization that is lower, about 165 mu per gram. And also the remanescent coercitivity of this sample is a bit higher than for the rest. As an alternative to this thermal annealing process, we designed an alternative process that was consisted of a microwave plasma annealing process in a homemade reactor that we can see here. This process has the main advantage that we only use 10 cycles of 30 seconds. All the process takes less than half an hour to make the reduction. That is like more than about 20 times lower than the thermal annealing process. And also, since the equipment is homemade, is a conventional microwave, this method we think can be easily scalable for industrial application. We perform some magnetic characterization of the synthesized nanoparticles by using this process, and we did synthesis with two working pressures, one about 0.24 millibars and then a second of lower pressure of 0.15 millibars, and we consider 5 concentration of hydrogen in the atmosphere. We see how the higher the hydrogen concentration, the higher the magnetization, the saturation magnetization we obtain. That can be expected. However, what was quite surprising to us is that the lower the pressure of the reactor, the higher the saturation magnetization of the sample. And we see how for the sample that we obtain the best results, the saturation magnetization is higher than 40 mg, which is a very promising result for this method considering all the advantages that can have in industrial applications. And we also see that when we increase the hydrogen content in the plasma, the coercitivity decreases while the remanence increases. We perform also some X-ray decharacterization, and here what is interesting is that we only see iron and hematite. We don't see any magnetite. We perform a fitting of the XRD spectra by considering a mix of iron and hematite, and we see an increase in the concentration of iron for the samples that have the highest saturation magnetization. This is consistent with what was expected, and we think that in this case, since the plasma is a very superficial effect, maybe we are only being able to reduce the surface of the nanoparticles. And maybe we think we have like a shell of iron and a core of hematite. We are only being able to reduce the surface of the nanoparticles, and this explains why we don't see any magnet. We only see the hematite and iron completely reduced. So as an application, we use these nanoparticles for cellular magnetic stimulation. For that, we used glial cells. We applied a magnetic field of 131 millitesla for 1 hour. We performed the experiments by using two frequencies, high one of 0.1 Hz and a low one of 10 Hz. We consider 4 concentration of nanoparticles from 25 to 100 micrograms per milliliter. And we consider both nanocubes reduced with the thermal method and the microwave plasma method, and also nanoellipses reduced thermally. And we measure the cell viability 24 and 48 hours after the experiment. Here we have the results, and what we can see is that 48 hours after the experiment, the viability of the sample that was microwave plasma annealed, or the cells that have these nanoparticles that were microwave plasma annealed, are the best one, better than the samples that were thermally annealed, both for low and high frequencies, and for concentration lower than 75 micrograms per milliliter, which is a promising result for this application of the microwave plasma annealed nanoparticles. So to conclude, hematite nanoparticles have been synthesized by using a hydrothermal method, and by changing the molecular ratio between the chloride and the phosphate, we can modify the shape and the size of the nanoparticles. We have been able to produce magnetic nanoparticles by applying a thermal hydrogenation process, and we have been able to obtain saturation magnetization of up to 207 mgrams. And by using the microwave plasma hydrogenation process, we have been able to obtain nanoparticles with a saturation magnetization of up to 41 mgrams. And we think we have a core of iron and of hematite and a shell of iron. And the applicability of the synthesized nanoparticles in reduced in cellular magnetic stimulation has been also demonstrated, and we have some work in progress that we want to see if we in fact have this core shell structure. We want to characterize our nanoparticles by yields. We also want to study the applicability of higher proportion of hydrogen in the plasma as well as lower pressures to see if we can increase the saturation magnetization, and we also want to functionalize the nanoparticles to improve their biocompatibility. So we acknowledge our funding, and thank you very much for your attention.
Okay. Thank you, Francisco, for the very interesting talk. We have time for one quick question. If anyone have, please unmute himself or put the question in the chat box. Okay, then if not, then okay, we can discuss something in the other time. Okay, Francisco, thank you. Thank you for the